And later, after lunch break, we'll have the keynote by the Cesare Fulanello from Italy with the topic that toward a scientific blockchain framework for reproducible data analysis. Because what is really a challenge now is to make data or the science reproducible again. And then we're going to have two talks, which are the proceeding talks by ISMB, by Georg Arneklevert from Bayer and Dat Duong from UCLA. After these two talks, the two proceeding talks, we will have the time for voting for the best presentation award. So you should have received, as a CAMDA participant, you should have received yesterday an email describing that you can vote for any of the talks which are presented, and you can pick at least one of them with giving it 3 points, but you also can nominate the second and the third with 2 and 1 point based on the quality of the presentation and of course the quality of what was presented. And after this we will count all the votes and pick the best best presentation, and the speaker of this best presentation will get the award, which is connected with here with the check with the Prague. After coffee break, we are going to have a session about the future challenges for CAMDA. It will be started by the talk by Wenzhong Xiao from Harvard Medical School and Stanford Genome Center. And later we are going to have a panel discussion where we would like to ask you as a CAMDA community what you find interesting, what kind of the questions you would like to answer, what kind of the data you would like to play. So if you have any specific interest or any specific interesting data set, then also please contact us so then we can prepare the summary for this panel discussion. And after panel discussion there will be a ceremony of nominating and giving the CAMDA trophy, and then we close with the remarks and with summary how we think that the CAMDA will be next year in Chicago together with ISMB there. So we will start with the talks based on for the MetaSub Intercity Challenge, and this session will be chaired by Chris Mason, who has started the MetaSub. Okay, so Chris.
All right, good morning, everyone. Thank you all for coming this morning, and it was an exciting day yesterday to learn about really complete, we saw viral genomes that were being populated across different surfaces and cities. We've seen many other sort of potential forensic applications of metagenomics data, which we'll talk about also for the future challenges. And before I introduce Moreno Zolfo to come give the first talk, and also to stay on time, we're trying to keep on time. I'll talk just for a couple minutes about just the background of MetaSub. So it began originally as actually a, there really, as most of you know, at least personally I can tell you how I got interested in metagenomics, and that is every time you sequence a human genome, most of you, all of you have probably taken your BAM file, looked at reads, you map to your genome of interest, you always have fragments left over. This is an inevitable consequence of living and having a body that is comprised of many kingdoms of life. So bacterial, viral, fungal, and some of you roll around in plants sometimes for various reasons, cornfields, what have you. We all are represented by sort of an ecosystem of cells, not just mammalian cells. And so I noticed for many years, every time we looked at a human genome, we would always have these other fragments, which of course today we all call just the microbiome or the metagenome. So I became, increasingly curious doing work in human genetics about what else was there. And then, of course, there was the International Consortia for Microbiome Research, the Human Microbiome Project from the NIH, which established clear colonization of different areas of the body and natural ecosystems that we all carry with us. And so, this had been brewing for the past really, 5, 6, 7 years, which was very exciting. But a lot of it was, driven by 16S research or 18S or ITS amplicon sequencing. And as sequencing costs became cheaper, it became much more likely and even feasible for people to do shotgun sequencing and look at all kingdoms of life whenever whenever you have a mysterious sample that you want to take a look at. And so we started tinkering with this a bit in the laboratory, and also there'd been a long-standing discussion of generating maps of cities or doing disease surveillance or microbial surveillance on a continual basis. There were some working groups at Google that was talking about making sniffers that would go on doors. So as you walk through doors, that you could have a puff of air and sequence the microbiome as people walk through every door at airport security. This does not happen yet, but one thing that does happen is thermographic cameras are already visualize your heat levels when you walk through airports to see if you appear to have an infection. And so if your body temperature is too high, authorities can and will move you to the side to actually, yeah, like there. So David will probably have to be quarantined shortly, I think. Gesundheit. So, there's been, over the past 10 years, all this discussion about what if we could be sequencing faster, more ubiquitously. Could we, Jana Verlek talked about this idea, the vision of ubiquitous sequencing, that you have sequences on doorways, on sewage channels, on airplanes. We just came from the MetaSub meeting in Stockholm, where Boeing is doing a project called the Fly Healthy project, where they are sequencing airplanes before, as people board the plane, and then in the middle of flight, trying to get everything collected with a goal to sequence in the middle of flight. Right now, they bring everything back to the lab, but they have a project as well that you could eventually have smart planes, you could have smart cities, you could have smart shoes, you could have smart laptops that can tell you what you've encountered. So there is a newfound excitement and really ubiquitous sequencing, many projects sequencing many things that we touch and interface with. And the MetaSub project was born out of that curiosity. A lot of it was with discussions with people who'd been doing this. For example, Eric Schott had done this at PacBio at the headquarters for the company, sequencing the refrigerators, the kitchens, when people got into work. And so a lot of it's been discussed for many years, but to my knowledge, the MetaSub is sort of the largest consortia for metagenomics, putting it together across many cities. And so it's the goals of it, which you'll hear some of the applications of the data, but the three main goals are to create metagenomic maps of the world around us, and also gives us some forensic potential, which we'll explore today, to discover and track also antibiotic resistance and antimicrobial resistance markers. And the third is a very clear discovery effort at biosynthetic gene clusters, so you can mine the data for biochemical function, as well as you can find new phages, new CRISPR elements. You can mine many things from the data because it's shotgun data. So those are the three main goals. And then I'll close with the thought that the final reason I became interested in metagenomics is just because I became a father, and I watched my daughter put things in her mouth and lick things and always touch things. And so what was sort of a brewing curiosity mushroomed into a very tentative and tangible curiosity that I wanted to know what she was doing with what she was putting in her mouth. And it turns out no one knew what was actually on subways, for example, so we decided to start looking. So it's a bit of the backdrop about the consortium and my laboratory's work to help get it started. And so without any further ado, and to keep on time, I want to introduce Miranda Zolfo from University of Trento, talk about strain level diversity in the MetaSub datasets, and kick it off. Thanks.